Morning. I'd like to welcome you to the first session on the EdaHealth section of this week's conference. And it's my pleasure to introduce our first speaker, our keynote speaker, Professor Martin Green from the University of Nottingham, who, after a period in practice and still in practice now, moved to the University of Nottingham 8-9 years ago now to focus on research in cattle health and production, but particularly in mastitis and milk quality. Before we start, I'd actually also like to remind people to put their mobile phones on silent, preferably switch them off. And if you have any questions at the end of the session, because the whole session is actually being recorded, could I ask you to move to one of the microphones in the aisles so that your question can be heard both by the speaker, but also be recorded for later broadcast. Thank you. Martin, the floor is yours.
Thank you very much, Andrew. So, good morning, everybody. Thank you very much for coming this morning. It's very nice to see you. I'd particularly like to thank the organisers for inviting me to speak today, and also for letting me speak first and then enjoying the rest of the conference. I'm sure it'll be a great week and everyone will enjoy the delights of Dublin. So my talk this morning is about mastitis control. I probably should apologise right from the start because this isn't going to be a talk about easy take-home messages and practical tips for how to implement mastitis control back on your farms. It's more going to be a gaze into the future and thinking about the sorts of things that might happen. And in particular, I'm going to be trying to make a case that predictive biology may provide us with a future in mastitis control. So, oh dear, we've lost a bit of formatting on the screen already. Again? Yeah, thanks. So what I'm going to do through the talk is very briefly look at the background of mastitis, think about what we do now, and maybe think about why and how we can get better at mastitis control, and then spend most of the talk thinking about what the future might hold for mastitis control. So it is tempting to think, isn't it, that we have cows, we have dairy cows, we're always going to have mastitis. And there are trends really going on around the world in the global dairy industry, increasing herd sizes, whether those cows be indoors or outdoors, probably reduced labour per cow being milked. Certainly over the recent period, we've had a very difficult time in terms of milk price, so income. So that's put a lot of pressure on herd health and disease prevention. And so the traditional methods of mastitis control that we've probably had over many years, that we've been developing over many years, of course will remain with us, but it does raise the question as we move forward into this kind of era of dairy farming, what does the future hold? So what might mastitis control look like, probably in addition to the rather traditional methods we use at the moment? I'm certainly not going to even hint that there's anything wrong with the basics of mastitis control. They can get us an awfully long way. There's a whole series of plans and schemes, and all of these deliver a really good job. So absolutely fine, we need to do these things. Mastitis control, as it is very important. But actually, what's going to happen next? Where are we going to go? What kinds of things are going to happen to mastitis control? Maybe before we do that, we should just dwell slightly on our current approaches. How do we go about mastitis control at the moment? And I guess I've split this up into two main areas. There's the sort of investigate mastitis approach. That's to say, when a problem arises, we maybe go to a farm and look at records and take some samples. It may be a somatic cell count problem. It may be a clinical mastitis problem. And we conduct some kind of an investigation with the details and the information and the management practices which are in front of us. There's then maybe a slightly more proactive approach, if you like. I've called it a herd health approach, where instead of going to investigate a problem, we're involved with the farm over time. So we're monitoring data regularly, evaluating that data, highlighting problem areas from the data and doing that relatively quickly, assessing the management, hopefully then identifying areas to implement changes and go back to recording data again. And again, although it's reactive in the sense that we're we're monitoring data and things have to happen to the data, nonetheless, we're hopefully on top of mastitis situations relatively quickly. Of course, an important element of mastitis control, and this was, I guess, largely raised by the Dutch Udder Health Programme, and a variety of research has been conducted in this area and remains being conducted in this area, that the importance of teamwork and communication and trying to elicit behavior change on farm, we recognize is a crucial area if we're going to achieve anything in mastitis control. So it's not good enough just to know technically what to do. We have to be able to encourage the team on the farm to be able to do that. And I would recommend to those of you who are interested in this area, Chapter 2 in this book on dairy herd health, which is written by Helen Higgins, is a really interesting look at behavior change, understanding human behavior change, taken from actually the medical literature to some extent. And it's a really good read if you're interested in trying to elicit behavior change on farm. So, they're all good. The things we do at the moment can take us quite a long way. But what kinds of things might happen next? What might mastitis control look like going forward? And I'm going to try to make the case for predictive biology. So predictive biology, essentially here, I'm going to call an understanding or knowledge of basic biology, biological processes and biological mechanisms to make forecasts about disease and its control, in this case about mastitis. So here we're interested to know if by understanding the basic biology, can we not only determine what's happening now, but can we in some sense predict or forecast what's likely to happen in the relatively near future so that we can make changes even in advance of them happening. And it's this predictive or forecasting phase which has the potential to make things even better for us in the future. So of course there's no shortage of data. If we're interested in data and making predictions, data literally are all around us, potentially on dairy farms. So we not only have the basic data of incidence and prevalence of mastitis, somatic cell count records, and so on. We can get data about the cow's environment, whether that be climatic data, it might be data related to crops and forage, it might be data related to bedding, such as dryness of bedding. Of course, because cows are milked 2 or 3 or more sometimes times a day, we can get all sorts of markers and data from the milk as it comes through. Increasingly, we have technologies associated with behavior on farm, so location monitoring is now something that we can do. Activity monitoring is something we can do. We can get a whole rounded understanding of the cow and her environment. We have much more information about pathogens, and that's an area which is evolving very quickly, and I'll touch on that a little bit later. And then, of course, we have information about the cow's genetics as well. So we have a huge amount of data which we can use to try to develop some predictive science from. And I guess that's the challenge for us. Can we put these data together in a sensible way? And then, of course, handling big data these days is not so much of a problem. So computing, computer power, the ability to synthesize and analyze data is really quite straightforward. So we've moved on hugely in these areas. At the moment, we don't really make the most of these in our day-to-day disease control, you could argue, but nonetheless, the opportunities are there. However, if we're going to be able to use data to make predictions and for predictions to be useful, then I would argue quite strongly we do need to be able to understand the underlying biology from which the data come. And probably too often at the moment we have data without understanding exactly what it means, biologically what it means, and how we can use that to make a difference. So I would also argue that we need to be able to create relevant questions which we can use the data to answer, but also to do it in a smart way. That's to say, to use the data to answer questions which will really help us to change the course of events. So help us to make choices about management and prevention and to make a real difference. So I guess the overall vision for this aspect is something like this. Can we synthesize data? We've got multiple sources, multiple formats, and actually combine that to have an effect, in this case, on mastitis, to be forecasting what might happen and to make a difference in terms of mastitis. And essentially what I'm going to do for the rest of the talk is to give 3 examples, is to use 3 different areas of examples of predictive biology. I mean, predictive biology is really going on in many, many areas. And it's a massive set of sciences, really. So I'm going to take 3 areas. I'm going to look at one surrounding the cow and cow susceptibility, one more to do with the pathogen and look at how subspecies differentiation of pathogens may help us to make some predictions about their behavior, and then go on to a more sort of numeric item involving modeling and looking at how some probabilistic models can also help us in terms of making good clinical predictions and looking at different contexts. So I'm going to go through these areas one by one and start off with cow liability or susceptibility to mastitis and how understanding the biology can maybe help us to make predictions for individual cows. I guess one of the most developed areas in terms of cow susceptibility is to do with the genomics revolution. So I think we're now well moving on through the field in terms of being able to understand elements of the genome and how they relate to liability or susceptibility to mastitis. There's been a variety of studies in this area where either particular gene markers or single nucleotide polymorphisms or SNPs through the genome are used to try to predict an individual's liability or susceptibility to mastitis. In terms of somatic cell counts, we have some reasonable information on this. In terms of clinical disease, we've probably got some way to go. And what's interesting in terms of the genomic evaluations are that the studies tend to suggest that mastitis is going to be a highly polygenic trait. That's to say there's going to be a whole lot of genetic areas which probably have some influence on mastitis, and so making predictions from the genome is going to be not completely straightforward, although certainly something that ought to be achievable. And no doubt these will progress. And essentially what we're talking about here when we're thinking about susceptibility or liability to mastitis is, of course, using the genome and genomic markers in terms of bull selection, and I guess that's the way that's very much accepted. Then moving much more towards looking at individual heifers and looking at maybe selection methods of young stock on farm, whether we're selecting against particular genetic patterns or for particular ones. And then I think this technology, of course, will move on and become maybe even more useful when we can make selections even earlier. So when we can start to look at maybe early embryos and look at conduct genomic evaluations at the embryo stage and make choices maybe in terms of embryo transfer technology of selecting animals with specific traits, including mastitis. So I guess as an example of predictive biology, this is the area which we would accept and know about, and is a good illustration of how, by understanding basics about the genome, we can maybe learn more about susceptibility or liability to mastitis. But of course, cow susceptibility is much more than just genetics. There's the effects of the environment as well. There may be direct effects of the environment, or the environment, of course, can have effects through modifying genes in so-called epigenetic effects. And another area that I'd just like to touch on here, because for me, I find it a very interesting area, fascinating area. I think an area we will see much more of. And I think we're also going to have another talk on this subject in a couple of talks' time later this morning. And this is the microbiome, in this case, the mammary microbiome. So the microbiome here is essentially the community of organisms, of microorganisms that we all have in our bodies that share our body space. And over the last few years, there's been more of a realization that these microorganisms actually have quite an impact on us, an impact on our health, an impact on our immunity, and therefore are quite important. And so in terms of the cow, you can see, we can understand that the mammary microbiome may well play quite a role in an individual cow susceptibility to mastitis. So in the human literature, and this goes back to 2012, this paper, but again, it interests me because the title was actually a very clever one and quite a good one. So the human microbiome, our second genome. So these workers in the human field were trying to make the case that, okay, we know the human genome is hugely important, and if you like, is a blueprint for our biology, but actually that they consider that our microbiome, the microbes that we live with, are of equal importance, and understanding those is just as important as understanding our genetic code. So they go on to say in this paper that the human microbiome is, amongst other things, a modifier of disease, an essential component of immunity, a functional entity that influences metabolism and modulates drug interactions. So if that is true, and this is clearly what the human research is going towards, it would be make good sense if this was actually had some fundamental influences on mastitis in cows. So our understanding of the microbes that live in the udder and probably beyond in the cow may be very important. So the mammary microbiome study, there was an initial mammary microbiome study in humans, so in breast milk, and this is that publication. So again, a few quotes from that paper. They found the communities to be very complex, a collection of bacteria which was much more diverse than had previously been thought to be the case. But they asked kind of like, in many ways, the obvious question and the fundamental question, but what do they mean? Why are they there? What are they doing there? And I think that, of course, that raises a lot of interesting questions. I think this was the first study carried out in cows by Okonoma and others, a cross-sectional study looking at the microbiome in cow's milk. So here we're looking for this microbial community as a one-off event in cows, in a variety of quarters of cows. And in this research, the workers divided the cows into healthy and subclinical and clinically affected quarters, and also the quarters were categorized according to somatic cell count level. And even just the graph here, which you're not obviously intended to read, shows that there's a spectrum of different genera of bacteria in these different groups of quarters. One of the interesting and key findings that came out of this study was that when they tried to discriminate between the microbial communities, even of the healthy quarters, then they did find differences between those quarters. So that's to say, quarters with a very low cell count or a medium cell count or a slightly higher cell count, but all of which were deemed to be healthy, you could identify differences in their microbial community, in the microbial makeup of those quarters. And again, it does raise the question, is that by chance? What does that mean? Is that cause or effect? Because this is a cross-sectional study. So is the difference in microbial community, does that occur because of the different sort of cell count levels, if you like, or does it actually mean something physiologically? So quite a few questions from where we are at the moment, and there's a variety of groups working on this in different parts of the world, the microbial community of the udder. But does it influence the susceptibility to mastitis? If so, in days of reducing antimicrobial usage, looking for alternative ways to enhance cow immunity as such, or reduce susceptibility to infection could be really important. Does the microbial community change over time? Again, we don't know we have no answer to that. How is it formed in the udder? Are there genetic or environmental influences on it? There's some information about that, but largely we're still not sure. Probably quite importantly, do we disturb it? And to what extent do we disturb it with things like antibiotic dry cow therapy and post-milking teat disinfection? So what do we do to this microbial community in our day-to-day management of cows? And I guess ultimately, in terms of the talk here, to what extent could we use this to predict a cow's susceptibility or liability to infection? So to answer this, and I think we might hear one such study, we need some studies over time. We need some longitudinal studies. What we really need is to see how this microbial community changes throughout a cow's life. and then relate that to maybe mastitis or susceptibility kind of events. Particularly of interest would be looking at that across the dry period, because of course the other changes fundamentally in that time, and also to a large extent there are a large number of cows which receive antibiotic dry cow therapy at that time. I think just to make us think a little bit more about this and to bring this just for a couple of minutes into a clinical kind of position, this is a drying off list of a 700 cow herd which has a somatic cell count, herd somatic cell count of about 100. And these are the last 3 somatic cell counts of cows which are about to be dried off in this herd. So the cows highlighted in red here, you can see have very low somatic cell counts in the last 3 recordings before drying off. And actually this isn't that unusual. If you do take some time and have a look at somatic cell counts before cows are dried off, you will see that quite a lot of cows have really quite low, maybe between 20 and 70 in terms of their somatic cell count. But it does make you wonder, if we're using blanket dry cow therapy, quite what happens in these cows, what happens in terms of the microbiomes of these cows if we give antimicrobial agents for a prolonged period through that dry period. And actually, if we don't do that and we use some other routes, including teat sealants, does that in some way preserve or maintain the microbiome? So we haven't got answers to these questions, but an interesting area and a good example of where, if we can understand that basic biology, it's a field that has a great potential to move us forward, probably significantly in terms of mastitis control. Okay, so cow susceptibility. I'm going to move on to a bit of research now on strain typing or subspecies differentiation of mastitis pathogens now as the second example of predictive biology. So here we're talking about, can we understand the pathogen biology, some basics of pathogen biology, to give us an insight into some important characteristics, some clinical characteristics of the pathogen, which can then help us to inform the control measures that we need to implement. And here in particular, we're going to talk about a bit of research which is being conducted at Nottingham. This was largely done by Piers Davis in his PhD, and has been making the differentiation in particular between contagious and environmental transmission of Strep uberis. So round about probably just over 10 years ago now, Ruth Zadok's published some very nice work around the contagious spread, potentially contagious spread of Strep uberis, and in one case an outbreak of what appeared to be contagious transmission of Strep uberis. And there's been various other work involving strain typing, which suggests that Strep uberis can sometimes behave in a contagious manner and spread between cows, as opposed to an environmental manner where infection is probably from the environment. And so it would be a useful differentiation to make if we could understand and differentiate strains of Strep uberis. That would help us in terms of the management that we need to implement on farm, because clearly the control processes are going to be different. We're going to be looking much more at parlour-type procedures, we think, at least for the contagious spread, as opposed to the environmental hygiene side. So if we could make this differentiation as an example of something which might be useful by understanding the biology of Strep uberis, then that could inform our approach to prevention. We would need to be able to do this quite quickly, and ultimately we'd need to be able to identify, when we found a clinical or subclinical case of Strep uberis mastitis, we'd need to be able to answer the question, if we could, well, which type is this? Because then what's likely to happen in the future? Again, a good example of how understanding something now would hopefully differentiate and determine a future event and our approach to control. So I'm just going to briefly mention a study which was published just a few months ago, which was taken from Strep uberis isolates from 52 herds over a year period. These are all clinical cases. We didn't have any subclinical mastitis in this particular study, which would have been very nice that we didn't. And the strain typing of Strep uberis was 2 methods we used. One is called multi-locus sequence typing, MLST, and the other is called MALDI-TOF MS, which I'll come back to in a little while. So essentially, multilocus sequence typing is a method to, if you like, genetically strain type Strep uberis. So 7 fairly conserved genes are fully sequenced, and depending on the sequence of each of those genes, they're basically given a number. And then according to how each of those genes is numbered, you give the particular strain type an overall number, or if you like, think of it like a barcode, which is unique to the particular sequences of those 7 genes. Because they're reasonably well conserved, this will differentiate the strains reasonably well, and we followed in this research, we followed the original paper, which was by Coffey and others, which was about 10 years ago, describing MLST. So we might have a separate strain of Strep uberis, which here varies at 3 of the loci, and therefore we would have we could determine that this is a different genetic type, but we could also identify to some extent how related it was to the one on the previous slide by how many loci vary using the MLST scheme. So what we were trying to say here was, when we were looking for clinical mastitis in different cows, we were wanting to know to what extent is this, how often are these the same strain passing between cows, and if we found the same MLST sequence in one cow and then a period of time later in another cow, that was evidence for a contagious transmission, or we called that at least a potentially transmissible event. I have to say, we did actually then do some full genome typing on these, on a subset of these Strep uberis isolates, just to really make sure that what we thought were the same strains, and in fact that turned out to be the case. So this is a graph that describes some of the results of that study. So these are the 50 farms along the x-axis, and they're basically colored by using 2 different bars. The number of cases of clinical mastitis is on the y-axis, and the 2 different bars represent cases of Strep uberis mastitis which were either caused by an event which was where the 2 strains between the cows were the same, so a potentially contagious type event, which are the dark blue parts of the bar or black parts of the bar, or the lighter colored bar below, which are where the strains were only identified once, which we were then placing as the environmental strain. So 2 types of strain here. So the darker bar, the contagious strains, and the lighter bar, the environment strains. And you can see a few things straight away just by looking at the graph in a very simple way like this. First of all, you can see quite a lot of black. So we actually found quite a lot of the same strains of Strep uberis were moving between cows on these farms. I'll give you some figures on that in a minute. You do find farms like this, where there was no Strep uberis, and a couple down here and one over here, where there was none of these potentially contagious events. And then you have some farms like this, where there was apparently a lot of the same strain of Strep uberis moving between different cows. So just to give you a couple of numbers on that, so in fact, 33 of the 52 herds, so around about 2/3 of the herd, had some evidence of contagious spread. So we think contagious spread was likely to be going on in that number. And possibly, slightly more interestingly, on about 1/3 of the farms, it accounted for more than half of the Strep uberis mastitis that they had. So in round about 1/3 of the farms, contagious spread may be the predominant route by which Strep uberis clinical mastitis infections were occurring, were being caused. When we look at how related the strains were, again using the multilocus sequence typing, and this is a blast analysis, the details are not important, but what is important is to say that the potentially contagious strains, the strains that were turning up in different cows in the same herd, were much more closely related than the environmental strains. So this group in the middle here are the contagious type strains. And this really, the point of this is just simply to make the point that it looks like their underlying biology may be more similar than the environmental ones which are scattered around the outside here. And if these share some common biological components or factors or basically some common biology, then it may be we can identify that biology and differentiate that biology so that we can then go on to predict when we find a Strep uberis how it's going to behave on the farm. And that's what this is about, is predicting, see if we can predict the behavior of a Strep uberis isolate when we first identify it. So can we do that? The problem with MLST and the analysis I've just shown you is it's a clunky research-based technique. It takes a long time. So what we really need is some method of doing this much more quickly. Can we isolate a Strep uberis? Can we conduct some tests on those? And can we package them and say, yes, this is likely to be a contagious strain, and this is likely to be an environmental strain? So we took this a little bit further using, whoops, wrong one, using this technique called MALDI-TOF-MS. So this is a technique which is based on typing Strep uberis using the protein profile of the organism as opposed to a part of the genetic code. So this is essentially a mass spec machine which separates mainly the ribosomal proteins based on their mass and charge, and you get a fingerprint which looks something like this, which we were hoping would be linked to that particular strain, and that we could differentiate strains according to their protein profile. So the question now is very similar, but we're now looking to discriminate our contagious and environmental populations when, in this case, a clinical mastitis crops up. Could we actually use these protein profiles to say, oh, yes, this one has probably come from the environment, or, oh, no, this one's probably come from another cow in the herd, and therefore help to inform our preventive measures? So to do this, to try to identify groups of peaks, one of the things that we did was carry out some principal components analysis. So principal components analysis is essentially a technique where we're looking for the patterns of the peaks, which peaks are present in one group compared to another, and if you cluster those peaks together and you make some models of clustering those peaks together, can you, or how well can you differentiate these particular two strains? And actually, it turns out you can do this really quite well. So this is one particular farm on the graph over here, and the red dots here represent Strep uberis, which, according to our MLST scheme, has moved between cows, so the contagious type, and the green here are the environmental type. And actually using the PCA type analysis, and this just gives our best 3 components here, but we can use more than that in our models, you can actually differentiate these really quite nicely. So theoretically, if a new Strep uberis came in this particular herd, and it was somewhere in this region, it may be, we could be quite confident that was going to behave in a contagious way, or has the ability to behave in a contagious way, as opposed to being an environmental pathogen. And there were 5 farms that had enough Strep uberis for us to do this in, and actually, they were all very similar. You could differentiate really quite well. Sorry, these graphs aren't quite as good. You see the red and the green being differentiated here. And when we look at these overall, and we put some numbers on that, and we look at an accuracy using cross-validation, so this is a technique where you miss some of those out and try to predict where they are. It turns out that roundabout with a 95% accuracy, you can predict which group they are on, or which group, sorry, they are from within these herds. So it has very good potential to be able to separate contagious and environmental strains. Interestingly, just behind this, when you look at those strains, the contagious strains in the different herds, and here we have an example of 4 herds, actually within each herd, you can discriminate them well, but between herds, they're also very different. So it looks as though the population of Strep uberis are evolving slightly differently in different herds, even within those that behave in a contagious way. We also have gone on to do some modeling about predicting future risk from these, And interestingly, again, in herds where a contagious type of Strep uberis turns up, indeed there is about a 2 to 4 week period where the cows in that herd appear to be at a greater risk of that spreading, of a new clinical case caused by a contagious isolate. So a good example, by understanding the biology, trying to make predictions of how the pathogen might behave, we can hopefully use that in the control of mastitis. And although we've talked just about that one area of contagious and environmental, I've just picked one phenotype because we're interested in it, there's all sorts of phenotypes that might be interesting here to do with antimicrobial resistance, for example, treatment outcomes. If we can understand the differences, the biological differences in these, then we could use all of these outcomes for us to make such predictions. Okay, so got nearly 10 minutes left. 8 minutes left, okay. So the last one I'm just going to mention is to do with using stochastic models to make clinical predictions, something that's actually quite close to my heart, somebody who likes statistics and epidemiology, but actually a technique I think we could use a lot more, and it'd be very useful in research if we could use a lot more, and I'm just going to describe it a bit briefly. So the motivation for this is really that for research to be useful, it needs to inform clinical decisions, and clinicians need to be able to understand that research in the sense they need to be able to put it into the context that they will see it in different herds. We need to understand how uncertain the results are, but particularly because research is done on certain herds in a certain context, we need to be able to say, well, if we move that to a different context that we know about, how applicable will the research be? And the kind of things the reasons to use these kind of models are because we can conduct a lot of what-if scenarios very quickly and examine these contexts, as I'll show you. We can combine data from different studies, which I'll show you a little bit of. Probably importantly, what we can do with these is try to find where, if you like, what we call the most important or the most sensitive inputs. That's to say, when we're looking at some context, we can identify which parts of management, for example, are the most important for us to address. And I'll just explain that a bit more. Okay, so what is this kind of modeling? What do I actually mean by this? It's used a lot in human medicine, particularly in randomized controlled trial analysis. It's essentially a way of considering lots and lots of contexts and combinations well beyond what we can do by experiment. So I'm just going to do a fairly simple example of this to kind of make the point. So suppose we have a case of clinical mastitis, which we are treating, and we want to know the cost effectiveness, if you like, of that treatment. And this may depend on a few things. It may depend, for example, on the bacteriological cure rate. It may depend on, for example, how much this cow transmits to another cow, the risk of transmission. The bacteriological cure rate, for example, may depend on lots of things. It may depend on the kind of pathogen involved, whether it's Staph aureus or Strep uberis. It may depend on the age of the cow. It may depend on a variety of things. And so this could represent a whole series of values, as could the transmission risk, as could, for example, the milk price. And so what we want to do is to be able to look at all the combinations of these different factors that make up, in this case, the cost of mastitis. And so we might do this by sampling from each of these distributions and sampling each of them one by one. And then for each of these samples, calculating the cost of clinical mastitis. And then if we can do this thousands and tens of thousands and even hundreds of thousands of times, you can see by sampling from those distributions, by plucking lots and lots of different values and combining them together, in the end, you'll get nearly every combination. So you'll actually be able to combine those so that you can get every combination, every context, every version of how those factors affect the cost of mastitis. Of course, there's many more than just those three things that make up the cost of mastitis, but that doesn't matter. We can do this with as many parameters and try to make predictions using as many parameters as we want. What sorts of things can we get out of this? Well, these are the kind of outputs, and again, shows the use of it. So we might take one particular parameter, in this case the rate of transmission, and see how that varies with the cost of mastitis. And we might get a graph like this. And of course, this cloud represents all the variability in the other parameters there are. But in general, we can see that the rate of transmission, as that increases, the cost of a case will increase, as you'd expect. But we can also start to do things like this, which is rank the amount of we can rank the different parameters and how important they are in terms of the cost of mastitis. And finally, we can also look for particular scenarios. So we can actually look to see, well, why is it that sometimes the transmission risk is high and the cost of mastitis is low, and vice versa? What's going on those particular farms? We can also take something like on-farm culture and also look at lots of scenarios here. And a paper is going to be given by Pete Down later this morning, actually using this technique to describe that. What I want to do quickly in my last 3 minutes is to go on to the very final question using this technique, because it's a clinical question that I think illustrates the technique quite well. So the clinical question here is, how much does mastitis on a unit influence the fertility performance or the reproductive performance of a unit? And so I want to use the technique to try to answer this question. So there is lots of research that mastitis does affect fertility, a whole bundle of research that clinical mastitis and somatic cell counts can affect fertility, but this is mainly of an individual at an individual cow level. So what we'd like to know is, to what extent does that affect a herd mastitis? Sorry, the herd reproductive performance. And to do this, we'll need to take into account uncertainty in our effect sizes, the variation that there is in mastitis between units, clearly, but also, importantly, the relative importance of other factors that affect fertility. So to understand how much mastitis affects fertility, we have to put it in perspective with other things. And this has been written up in a paper by Chris Hudson, this technique. And essentially, I'm not going to go into the details of it, but you can create a model with different data sources. And then what we need to do is to create the hundreds of thousands, or even more than that, herd scenarios, which we've talked about, where you look at herds with different levels of reproductive inputs, such as heat detection and pregnancy rate. You look at herds with different mastitis performances, and you can then look at the relative importance of these different outcomes. So again, some example results. And again, what you find here is quite nice because it kind of makes a point. So the things that you would expect to affect herd performance a lot, and I've picked out one, which is the first service submission rate here, it does seem to. So as first service submission rate or heat detection increases, the financial losses of fertility are reduced. But if we look at the clinical mastitis here, there's not quite such an obvious pattern. And so, in fact, when you look at the relative importance here of clinical mastitis as opposed to other things, although we know, and there's lots of research to tell us, and we use the input parameters from those research, that mastitis does affect reproduction in cows, when you actually put that against other factors that affect reproduction, it turns out there are very, very few scenarios where clinical mastitis or clinical and subclinical mastitis on a dairy unit has any meaningful impact on its reproduction. And I think that's where this technique can be really useful in trying to understand how, even though there's a dozen or so papers which suggest that mastitis does affect reproduction, and at the cow level it certainly does, when we put that into a wider context of the herd situation, then in fact it's very small. So these sort of models are useful for making predictions, and a different illustration of how understanding biological processes, but combining them in a wider model, we can start to make predictions and particularly identify scenarios where either management needs to change or factors are important or less important. So I'm just about done now. I've tried to, admittedly, probably race through a little bit, a few areas of predictive biology, really to give an illustration of why I think that this is an area that will be important for us in the future. By using underlying principles, by understanding biology in this way, I think we will be able to move on into a new era of mastitis control, which will be much more predictive than where we are now. And there's huge opportunities in terms of the data and the sources available. So I would just like to thank my colleagues and co-authors very much. Lots of people were involved in the thoughts behind the talk and our funders. And I'd also just like to thank our wonderful group at Nottingham and look forward to having a beer later. Thank you very much.
Thank you, Martin. We've got time for perhaps just one or two quick questions. If you have a question, I could just remind you to move to the microphone in one of the aisles. Do we have any questions? Martin, I'd perhaps have one quick question, I'm afraid. And actually, you talk about the stochastic models and the predictive biology. How do you find farmers deal with actually those sort of discussions if you take them to the farmer and actually deal with, rather than a straight yes-no answer, some probabilities around the likelihood of outcomes for treatments and interventions on their farm?
Yeah, okay, thank you. I mean, good question, and you probably picked up, I deliberately kept this quite a long way away from the farm, this kind of stuff, but nonetheless, it needs to get back there. So I think our experience of that is actually they deal with that a lot better than we might imagine. But of course, I think the real answer is you have to decide that farmer by farmer. So there are some clients that you can work with that will deal with that very well, and you can talk about probabilities, and you can actually use them to make joint decisions. Whereas I also think we all probably recognize the fact that there are times when there are farmers where we may want to be working with more black and white looking scenarios. So there may be probabilities that we've got there, but in fact, we don't always portray it like that. So my feeling is that needs to be closely linked to the kind of farmer and situation that you're working with.
Thank you. One question here. If you could state your name and country, please.
Scott McDougall from New Zealand. Very nice talk.
Scott,
as usual, mate. How are you, mate? Yeah, just fascinated on that uberis data where you've got some herds where uberis is behaving very much as an environmental and other cases contagious. And I guess my question to you is, have you got enough herd level data to try and understand, is it the management of those herds that then allows uberis to behave in a contagious way, or is it something about the bugs that have ended up in that herd that then result in that contagious pattern?
Yeah, I mean, that's a really good question, Scott. So with this study, we have got quite a bit of herd data, and the truth is we did some modelling of herd factors, and you can't really identify. Now, that doesn't mean that the herd factor is not important. It may be we haven't got sufficient depth and richness of data to identify them. I think one of the things we do question is, so I mean, Strep uberis is slightly different to some other pathogens in that the concentration that it tends to be in milk is really quite high, and it may be that transmission is possibly easier actually than for some other pathogens, although we don't know that. But what I'm saying is I don't think we can be sure that the conventional methods of contagious control definitely apply, for example, to Strep uberis in the same way. But I think the herd issues around that, so I think the best guess is at the moment you do the same thing for contagious control that we think, including, post-milk contagious infection and so on, but actually, with the caveat, it may not be quite as simple as that.
Thank you. And with that last question, I'd like to draw this particular talk to an end, and perhaps you could join me in thanking Martin for his presentation.